Now let's talk about the drug discovery process. How does the drug is discovered? Start with identification and validation of a biological target. What is the target? Either the heart or the basal ganglia in the brain for Parkinsonism. You identify, or the gut, whatever, wherever you want to use the drug. You apply genomics and bioinformatics to this particular thing. You develop an assay and screen a large number of compound collections, which you've already got out of the computer. High throughput screening is the name of this computerized provision of drugs. And there's a robotic high throughput screening. This computer itself is under the control of a robot. So that will give you also a number of drugs. Compounds are collected, compound collection, and there are combinations, combinatorial biosynthesis and chemistry, which form some new drugs for you to use. Then you optimize some hit compounds. Hit compounds are the compounds you want to concentrate on to improve the efficacy, the safety, and the stability. Again, combinatorial biosynthesis and chemistry are used. Safety, PK, and pharmaceutics, PK and PD studies are used on these drugs. These are first used, of course, in animals. Then you've discovered the drug. After the animal has produced a single drug, which is fairly, immediately go for intellectual property. Intellectual property means you are going to stake your claim on this drug. Nobody can now come in the way of your research. Inputs from the market, meaning now we had a trial once on hemophilia. Hemophilia is a very rare drug, a very rare disease. And a very nice company brought out a recombinant type of treatment, which was actually a substitute for one of the blood coagulation factors. That drug was a miracle worker. We in JCDC tried it on several patients. I was involved in the trial, being a physician. And the patients even underwent surgery. Hemophilia, is a bleeding disease. You do a cut and the patient can bleed to death. So no surgery is possible. Patients came to us with fractures, with knee problems, with old fractures. They were operated upon and cured just by giving this drug so that no bleeding occurred during the surgery. So this is the good part of the hemophilia trial. However, this particular company went out of business. Why? Such a small amount of hemophiliacs and such a huge investment they had to make. And so it was not a blockbuster drug where it can be used in the whole lot of patients. The blockbuster drug is the statin, which I talked about last time, which brought down the levels of myocardial infarction, of hypertension, of cholesterol, which are a dime a dozen in the market. So the input from the market said, instead of making factor 8 and factor 9 in the laboratory, why don't you make a statin? You'll be able to use it in thousands of patients. So your inputs also have to be from business models and the market. Finally, there's a molecule of modeling. I told you about dextro and levo. You have to model the molecule so it gives the best result. Finally, the selection of the final candidate, which is going to be into trials. So this is the drug discovery process. You don't need to know all this, but you should be aware that this is the big process